Welcome to our presentation on corals and cryptic lineages.
So, the coral reefs, as an introduction, we want to talk slightly about why they are so important and why we are studying them. And so, one of the major things that coral reefs offer to the ocean is a nursery for a large portion of the marine life. They provide protection from water currents and allow fish to have a safe area and all the other marine life. So the issue that we're presented with right now is that climate change is causing warmer waters. And because of the warmer waters, these corals that provide all this protection are no longer able to provide the protection because they are bleaching or dying. So because of the destruction of the marine life, ecosystems is so relevant to our society, we're like, oh my gosh, we should take care of that. So one of the things that is saving the corals is the genetic variation and how they're able to slightly be a bit different, and then now they're able to adapt and survive in the warmer waters. Next slide. So our specific coral of interest is Orbicella. Our coral of interest is Orbicella fabulata. The samples that we got from Rachel's lab came from the Florida Keys National Marine Sanctuary and Flower Gardens Bank from both the East and West Banks. Now, you might ask, well, why these species? It's because they are under huge risking extinction, as well as possible cryptic lineages, which allow them to survive, which is of interest to scientists because we are able to figure out why it is that that cryptic lineage is able to survive better than the others. And we would be able to understand the dynamics of the ecosystem in the future. Next slide. So our goals and questions in this experiment were to find the clones in our samples based on identity by state. So one of the interesting things I didn't go over really quick is that these corals, when they have babies, they release their babies out into the ocean and they can swim. So they're in the water, so they can spread, which is why they're allowed to have cryptic lineages and change a little bit here and there. And that's why we're investigating east and west. But so then the biological question would be, are these clones cryptic lineages present in our data set? So we are looking for whether or not they were able to adapt and move around. And then our experimental question is just to find the genetic variants in these samples to support that.
So I'm going to talk about methods what we did. Here and like how the samples were collected. The experimental design here is that we use 5 colonies of corals, and those colonies, the samples of those colonies were fragmented into smaller pieces, and then RNA was extracted from the tissues. So we used the tag sequencing, and then cDNA was synthesized, and we extracted the RNA from cDNAs. Then we added Rachel added the adapters to cDNA, and the results then were sequenced with Illumina. Next thing is that the adapters and poly-A tails were trimmed and filtered for high quality reads, and the filtered results, we mapped them to then connect them to the coral colony, which is the Montastrea cavernosa, along with clade C and clade D genome. And then there are like a bunch of filters added to it. Loci were filtered with MAF below 0.05. And then we created 3 plots for this project. We got a dendrogram, PCA, and DPAC. So the dendrogram is that we use the proportion of loci for each sample, Orbicella fabulata and Florida Keys one, and we calculated the genetic distance, and then we use that genetic distance to plot the dendrogram. For PCA, we converted the SNPs data into principal components, and then those results were used to plot PCA. And for DPAC, we used the variance among population. We used the variance among population, partitioned variance among population into between population and within population components.
Okay, so for here's our dendrogram. It's very there's a lot in there, so I will zoom into it in a minute. But this, just from the get-go, you can tell that the FKNMS, the red portion, isn't really interspersed in the dendrogram when you compare it to the rest of the west and east FGB population. So when you zoom in, and before I get in, long lines in these dendrograms mean that these corals are genetically distant. So longer they are, the more distant, and shorter means they're not as distant. So when you zoom in on this purple circle, you can tell that these Zeds are known clones, so which means they are from the same colony, and OFAB 41, 19, and 32. So the black words and the orange ones are suspected clones. They're on the same branch with short lines. So they're the suspected clones. We're not sure yet. And the alphabets represent different clones in this dendrogram. And other zoomed in portions of this dendrogram is that one thing to note is that O frank and O fabs are different species of corals. So in the first picture on the left corner, the W's are the known clones because they're also on the same branch. The figure on the right corner is showing the known clone and the suspected clone. So the Y's are the known clones. And the very last one at the bottom, the OFAB Z15, that one is a suspected clone because it is on the same branch as the rest of the known clones. And this is a final zoomed in version of a portion of the dendrogram. So you can tell that V's and X, they are known clones because they are also from the same colony. And moving on to our PCA plot. So right off the bat, you can tell that the FKNMS, the red circle, it shows genetic variation on the first most variable locus, which is our PC1. And FGB shows genetic variation on the second most variable locus, which is our PC2. So FGB is spread out more, I guess, along the x-axis, and FKNMS is spread out more along the y-axis. So east and west banks overlap a lot, as you can see from the circle, which means they have similar genetic differences, and the west population has less variation. Okay, so when you look at the DPAC plot, this is derived from the PCA plot. So you can tell that FKNMS and FGB are clustered in different areas. The red circle is more towards, I guess, the right end of the square, and the rest are more clustered along the left area of the square. So this confirms our PCA plot that FKNMS corals and FGB ones aren't that related. And this can be also seen in the dendrogram where I mentioned that the FGBs are more interspersed along each other, while FKNMS are kind of clustered on their own.
The first thing we notice is that OFAV, FGB 41, 19, and 32 are likely part of the FKNMS Z clonal lineage, or that they are very, very closely related to that line of coral. Then we see that OFAV Z15 is actually most likely part of the Y clonal lineage. It might have been mislabeled. Finally, for the dendrogram, we see a possible cryptic lineage between FKNMS Z6, 2, and 5 and the rest of the FKNMS population. And we see that with Z6, 2, and 5 being on the top of the dendrogram and the rest of the population mostly on the bottom, which shows that there's significant genetic difference between these two groups. The FKNMS population and the FGB populations are most variable at different loci, which shows us that they're not very closely related to one another. So theories about the drift of coral polyps from FGB to the Florida Keys are clearly not holding up under this PCA analysis. We're not seeing a lot of gene flow between the two groups. Also, we can see two clusters as highlighted here with the green circles in the FGB population, which could be indicative of a cryptic lineage. However, it's also important to note that since two species were sequenced in this analysis, this could also just be species differences, and we'll need to do some more examination to determine if this is variation within the same species or variation between two species. The DPAC plot confirms that the FGB population and the FKNMS population are not very closely related. However, in this plot, we can also see the divergence of the FKNMS population as highlighted with these green circles. One possible reason for the formation of these cryptic lineages could be because, as we see, there is just a little bit of overlap with the FGB population, and so it's possible that some of those coral polyps are actually making their way to the Florida Keys and starting a cryptic lineage over there, but not enough to make the two populations very genetically similar.
So in conclusion, we did find several individuals to be suspected to be clones, as well as several cryptic lineages within the FGB. So we also see some clustering within the FKNMS lineages, like we've previously mentioned. And then, but the FKNMS and the FGB populations appear to be distinct. These results that we gathered can help us get a deeper understanding of the gene flow and migration patterns among these populations, just like Rachel mentioned with the larvae of the corals. For next steps would probably be collecting more individual samples, and in order to determine whether the cryptic lineages we mentioned are legitimate or not, as well as it can help us see the ecosystem changes that are driven by these cryptic lineages.